I want you to think about a specific kind of exhaustion. Like, you know the kind, I mean. You haven't run a marathon.
Right. You haven't been out lifting anything heavier than, like, a coffee mug.
Exactly. You might have literally just been sitting at your desk for 10 hours straight, right? Just locked into this intense, unbroken mental work.
Yeah, that deep focus.
But by the end of it, your head physically feels heavy. Like a literal fog just seems to roll in right behind your eyes, and you can barely string a sentence together.
We have definitely all been there.
Right. You just feel, I don't know, full.
Yeah, and it's a very physical reality, actually, because your brain is essentially this incredibly demanding engine.
Because it uses so much energy.
Right.
Oh.
Massive amounts.
Well,
think about the sheer physics of it. I mean, the brain makes up maybe 2% of your total body weight.
Yeah.
But it burns through 20% of your body's energy.
That is wild.
And it never stops running. Even when you sleep, it's just constantly humming along.
And so if there is one thing we know about engines, it's that burning fuel creates exhaust. It's exactly.
The brain generates this mountain of biological trash every single day. I'm talking spent proteins, metabolic byproducts, cellular debris, all of it. And here is the massive, like, century-old biological mystery we are tackling today. If a factory produces that much trash, it needs an industrial-sized plumbing system to haul it away.
It has to go somewhere.
Right. But the brain doesn't have a traditional lymphatic system. Like, it doesn't have those standard built-in drainage pipes that the rest of your body completely relies on.
Which is wild when you consider the stakes here. I mean, if that cellular debris isn't cleared out, the brain essentially just chokes on its own exhaust.
Yeah.
And for the longest time, the scientific community operated on this very specific set of assumptions about how this invisible waste disposal system worked.
But as it turns out, the old plumbing manual might be entirely wrong.
Basically, yeah. Completely wrong.
Okay, let's unpack this, because we are looking at a groundbreaking 2026 paper published in the journal Cell by a team led by Chayama.
Yeah, brilliant team. And it completely rewrites the map of the human brain. The paper is called Physiological Brain Clearance Architecture Revealed by Neuronal Protein Tracing.
Quite a mouthful, but very accurate.
Right. So in this deep dive, we are going to explore this fundamentally new understanding of where our mental trash goes, uncovering secret immune checkpoints, biological zip codes, and really looking at how a failure in this system might be the true origin of diseases like Alzheimer's.
Yeah, it really shifts our perspective, from viewing the brain's clearance as this simple mechanical drain to realizing it is a highly sophisticated, geographically organized communication network.
But I guess to appreciate what this new paper gets right, we have to look at the flaw in how scientists used to study this.
Yeah, the mechanical flaw of the old studies is key here.
So if I'm a researcher 10 years ago, right, and I want to see how the brain takes out the trash, what was the standard playbook?
Well, you would use a tracer injection. You'd take a fluorescent protein, usually something like fluorescent ovalbumin, or OVA, that glows really brightly under a microscope.
Yeah.
And you would physically inject it into the cerebrospinal fluid.
Okay, the fluid that cushions the brain.
Right. And usually they'd inject it right at the base of the skull. Then you basically just wait an hour or two and follow the glowing path to see where the fluid naturally drains.
And when they did that, it looked like almost all of the waste just rushed right past the brain tissue and dumped straight into the deep cervical lymph nodes in the neck.
Yeah, like water rushing down a shower drain.
But that's not actually what's happening.
Well, that was the accepted model for a long time. But there is a massive physics problem with that experiment. You see, the skull is not an open system.
Right, it's bone.
Exactly. It is a rigid, sealed, highly pressurized bone vault.
Ah, so the moment you pierce that vault and pump extra liquid in?
You're messing with the pressure. Precisely the issue. Even if you inject a tiny amount of fluid at a very slow rate, you are fundamentally altering the intracranial pressure gradients. You're changing the very fluid dynamics you're trying to observe.
Because it's suddenly overpressurized.
Yes. By forcing fluid into a closed, pressurized system, you create artificial pathways. You literally push the fluid through anatomical exits that might not actually be used for normal, everyday waste clearance.
Okay, if you're listening, try visualizing it like this. It's like researchers wanted to know how a busy factory takes out its daily trash, but their method was to dump a massive pressurized flood of neon water through the front door.
Right, just blast the doors open.
Exactly. And then they watch the water burst out the emergency exits and flood down the street into the storm drain, and they conclude, yes, this is exactly how the workers take out the recycling.
That is a perfect analogy. It's a flow artifact. The sheer force of the intervention creates a false result.
So how did the Chayama team fix this?
Well, that's why the innovation in this cell paper is so elegant. They didn't inject fluid into the skull at all. They developed a completely non-invasive genetic tagging system.
Okay, genetic tagging. How does that avoid the pressure problem?
They used a highly specialized, harmless virus, specifically a viral vector called AAV-PHP.E synapsin 1 Cre.
Wow, okay, AAV-PHP.UB. Got it.
Yeah, no, it's a lot of letters. But the cool part is they injected this virus into the regular bloodstream intravenously and just let it cross naturally into the brain.
And this virus, it acts kind of like a delivery truck, right? Like it only parks at the neurons, the actual brain cells.
Exactly. It targets the neurons and drops off a specific set of genetic instructions. It essentially tells the brain's own cells to start continuously manufacturing and secreting a highly stable, glowing green protein.
Which they call ZsGreen.
Yes, ZsGreen.
So there are no needles piercing the skull, no pressure changes. The brain is just naturally producing this glowing green waste product at its own normal physiological pace.
Exactly. It's completely natural flow.
But hold on, I have a massive problem with this. I can hear the skeptics already.
I know where you're going with this.
You're telling me they infected a brain with a virus to make it produce a glowing green alien protein. But how is a viral protein supposed to tell us literally anything about how my brain clears its own natural everyday waste? Wouldn't the brain treat that foreign protein totally differently?
That is the exact vulnerability the researchers knew they had to address. I mean, they had to prove this artificial ZsGreen behaved identically to endogenous waste.
Okay, so how did they prove it?
To do that, they used a technique grounded in click chemistry, a system they called Synfers.
Okay, Synfers. I've seen that term in the paper, but walk us through how that actually works in a living brain.
Okay, imagine the brain is an assembly line building proteins out of standard building blocks, right? Amino acids.
Sure.
The researchers engineered a way to slip a synthetic, unnatural building block, an AZF marker, directly into the mice's food.
In their food. That's crazy.
Yeah. And because of a genetic modification in these specific mice, only their neurons would pick up this synthetic block and use it to build their everyday natural proteins.
Wait, so they're basically sneaking a tracker into the normal supply chain.
Exactly. And this synthetic block has a tiny hidden chemical handle on it. The proteins still function perfectly normally. Then later, the researchers introduce a fluorescent tag that is chemically drawn to that specific handle.
So they click together.
Yes, they click together. So now they can map the exact drainage pathways of the brain's real natural proteins.
That is incredibly clever. So they track the brain's natural proteins using these chemical handles, and then they compare that map to the map made by the glowing green viral waste.
Yep. And the overlap was nearly perfect.
Oh, wow.
Yeah, the ZsGreen map mirrored the natural protein map flawlessly.
Oh.
They proved that ZsGreen acts just like the brain's natural waste, completely validating their new method.
Wait, before we look at the new map, let's make sure we have this foundation solid for everyone listening.
Good idea.
The old science told us the brain was a sink with a single drain leading to the neck. But that was really just because researchers were artificially overpressurizing the skull.
Right.
This new method lets the brain's own cells generate a glowing tracker at a normal pace without disturbing the pressure. And they've chemically proven this tracker perfectly mimics real waste.
So we finally have a clean, undisturbed view of the system for the very first time.
Okay, so what did they actually see?
Well, the researchers used a technique called 3D light sheet fluorescence microscopy, or LSFM.
That's where they clear the tissue, right?
Yes, exactly. They treat the mouse heads with chemicals that strip away all the lipids, the fats. It makes the skull and the brain completely transparent, like glass.
That sounds like science fiction.
It looks like it too. All that's left visible are these glowing green networks of ZsGreen waste.
And what did that glass skull show them? Like, where is the waste actually going if it's not just flushing straight down the neck?
It pools in very specific, highly compartmentalized regions. They found massive accumulations of waste in the dorsal dura, which is the tough membrane sitting right under the very top of your skull.
Okay, top of the head.
They also found it in the bone marrow of the skull itself, along the sides and the base, and they found it heavily pooling in the cribriform plate, right above the nasal cavity.
Wait, so it's not a single central drain at all. It's more like a sprawling city, and it looks like the waste is spreading out to multiple different borders.
It is, but it's not random. This is where they discovered what we can call the nearest exit principle.
The nearest exit.
Yeah, so they ran a variation of the experiment. This time, they genetically restricted the ZsGreen production to only specific, localized neighborhoods of the brain, rather than the whole thing at once.
Let me make sure I'm visualizing this right. If they only tell the forebrain, like the frontal top part of the brain to produce the green waste, does it still go all the way down to the nasal cavity?
No, that's the amazing part. Waste produced in the forebrain almost exclusively travels straight up. It exits into the dorsal dura at the top of the skull.
It uses the closest local exit.
Exactly.
Yeah.
But when they restricted the waste production to a region called the striatum, which is buried deep down in the lower central part of the brain, the waste completely ignored the top of the skull.
Let me guess, it went down.
It drained straight down through the base of the skull and out into the nasal cavity.
Okay, wait. If you're anything like me, you probably assumed your body just flushes out waste as fast as humanly possible, right?
People do, yeah.
I mean, if I'm generating biological trash, I want it gone, out of the system into the kidneys or liver to be destroyed. Why would the brain organize its waste by geography? Like, why route it to the closest local patch of skull instead of just using some superhighway to get it out?
Well, the researchers wanted to know the exact same thing. They needed to understand the kinetics, the speed at which these local dumps were operating. So they ran what's called a pulse chase experiment. Think of it like turning a faucet on, letting the sink fill, and then abruptly turning the faucet off to see how fast the water drains.
Gotcha.
They let the brain tag its proteins for a week, then shut the tagging system down, and measured the clearance over time.
Let me guess, different exits have different speeds.
Drastically different. The nasal cavity and the dura are fast-clearing routes. The waste clears out quickly. But the skull bone marrow, the areas bordering the brain, were incredibly slow.
How slow are we talking?
The waste doesn't just pass through, it lingers. It literally sits there for days, slowly degrading.
Oh, wow. So it's acting like a stagnant retention pond.
Exactly.
But why? Let's recap for a second because this is huge. First, injected tracers don't equal natural brain waste. Second, neurons use local neighborhood-specific exits. And third, the skull acts as a slow-moving retention pond for waste.
Perfect summary.
So if you're listening, think about your own daily routine. If you generated a specific type of trash, wouldn't you want the closest exit? But what stands out to you about the brain organizing its waste by geography rather than just speed? Like, why would the brain intentionally slow down its waste disposal right at its own border?
It is a huge question.
That feels like leaving bags of garbage on your front porch for a week. What is waiting in that skull bone marrow that needs so much time with the waste?
So what does this all mean? To figure that out, the researchers had to zoom in to the individual cells living in that slow-clearing border zone.
Okay, how do they do that?
They used a technique called single-cell RNA sequencing, or the cARNA-seq. It's essentially a way of isolating a single cell and reading its genetic diary line by line to see exactly what instructions the cell is executing at that very moment.
Oh, I love that. Reading the diary of the garbage worker. So who is reading the diary?
They found that local immune cells, specifically macrophages and B cells that reside permanently in the skull's bone marrow, were physically engulfing and eating the glowing green brain waste.
Wait, okay, so the local immune cells are eating the brain's trash.
Yes.
But normally, if an immune cell eats a strange protein, that means war, right? I mean, it sounds the alarm and triggers massive inflammation.
That is the standard immune response, yes. If a B cell eats a piece of a bacteria or a dead cell, it usually upregulates inflammatory genes, preparing the body to attack.
Right.
But when the researchers looked at the genetic diaries of these specific skull B cells, the ones full of the brain's green waste, they found a completely counterintuitive reaction.
They weren't sounding the alarm.
Quite the opposite. Now, they did turn on the genetic machinery required to chop up the waste and display it on their surface, like genes ARAP1 and PDA3. This is a process called antigen presentation.
Okay, so they are basically holding up a molecular piece of the waste like a wanted poster for other immune cells to see.
Yes.
But if they are holding up a wanted poster of the brain's own proteins, wouldn't the immune system attack the brain? Like, that's literally an autoimmune disease.
What's fascinating here is the secondary set of genes these B cells activated. At the exact same time they displayed the wanted poster, they heavily upregulated tolerogenic markers.
Tolerogenic.
Meaning they promote tolerance. They produced a massive amount of chemical signals, including a receptor called IL-10RI and a checkpoint protein known as PDL1.
Wait, PDL1, I've definitely heard of that in cancer research. Isn't that the cloaking device some tumors use to tell the immune system, hey, I belong here, don't shoot.
That is precisely what it is. It is a powerful biological stand-down signal.
Okay, let me try to put this into a visual because this completely changes how I view the brain.
Go for it.
These immune cells in the skull aren't border guards looking for an invading army. They are more like royal taste testers.
Oh, I like that.
Right. The brain is deliberately slowly feeding them a steady diet of its own natural garbage. And because this skull exit is a slow-moving retention pond, the B cells have the time to thoroughly chew up and display this waste while simultaneously broadcasting the stand-down signal.
Yes.
They are actively training the immune system to tolerate the brain's unique proteins.
You've hit on the core revelation of the paper. The slow clearance is not a biological design flaw. It is a vital immunological necessity.
Wow.
Think about it. The brain is walled off by the blood-brain barrier, right? So the general immune system rarely gets to see what healthy brain tissue actually looks like.
Oh, true.
So the brain carefully bags up its everyday waste, sorts it by neighborhood, and ships it to local bone marrow checkpoints to continuously update the immune system on what constitutes self. It literally prevents the body from accidentally launching an autoimmune attack against the brain.
That is absolute genius. The plumbing system is actually a high-tech communication and education network.
It really is.
But as any homeowner will tell you, pipes eventually break. I mean, plumbers make a fortune for a reason. What happens to this delicate immune training camp when the brain's plumbing leaks or clogs?
Well, the researchers asked that exact question. They took this newly mapped system and tested it against two distinct disease models.
Okay, let's hear them.
First, they looked at acute neural inflammation, which is a sudden, violent immune event. And then they looked at a chronic model for Alzheimer's disease. And what they found was that the system breaks down in two completely different ways.
Okay, let's start with the sudden violent one, the acute inflammation. What did they do to the mice?
They injected them with a bacterial endotoxin called LPS.
LPS, okay.
It basically simulates a severe whole-body infection. It causes a massive, rapid spike in inflammation.
And what happens to our carefully routed, glowing green waste?
Well, under normal conditions, that waste drains to the skull and nasal borders, right? But under acute inflammation, the delicate blood-brain barrier destabilizes. It becomes highly permeable or leaky.
Oh, no.
Yeah. The researchers found a massive spike of the green waste directly in the blood plasma, while the levels reaching the local skull checkpoints just absolutely plummeted.
So if the blood-brain barrier gets leaky, that the waste doesn't just spill everywhere, it completely bypasses the local skull training camps. Here's where it gets really interesting. It's like a pipe bursting in the basement. The water just aggressively dumps into the general bloodstream, and the local immune cells never get their delivery.
The strict geographical routing is completely lost. Now, compare that to the Alzheimer's model. They used mice genetically engineered to aggressively develop amyloid beta plaques in their brains as they age. It's called the 5XFAD model.
Okay, and just for our listeners, amyloid plaques are those sticky, misfolded clumps of protein that build up between neurons and are a major hallmark of Alzheimer's disease.
Right, so they track the clearance in these 5XFAD mice at 2 months old, and then again at 7 months old as the plaques grew.
And what happened?
Unlike the burst pipe of acute inflammation, the green waste didn't flood the blood. Instead, it just stopped clearing.
Stopped entirely.
Pretty much. The physical mass of the amyloid plaques trapped the glowing waste deep inside the brain parenchyma, the actual brain tissue itself. It couldn't reach the borders, and it couldn't reach the blood. It was just stuck.
Okay, so acute inflammation is a burst, leaky pipe flooding the basement, the blood. But Alzheimer's pathology is like a clogged drain where the sink just slowly overflows back into the brain tissue itself.
If we connect this to the bigger picture, you realize that both of these very different mechanical failures lead to the exact same disastrous immunological outcome.
Wait, really? How?
Think back to those tolerogenic B cells, sitting in the skull bone marrow, waiting for their daily delivery of brain waste to maintain the peace.
Oh, wow. If the waste leaks into the blood during inflammation, the B cells don't get it. And if the waste is trapped by plaques in the brain during Alzheimer's, the B cells still don't get it.
Exactly.
In both scenarios, the royal taste testers are completely starved of their samples.
The training camp is shut down. The border guards are suddenly deprived of the specific brain antigens they need to maintain immune tolerance.
That's terrifying.
And this sudden loss of communication might be the spark that ignites the dysregulated neuroimmunity we see in late-stage neurodegenerative diseases. The immune system literally forgets what the healthy brain looks like, and the tolerance breaks down.
Let me pause and try to synthesize this, because this profoundly shifts how we understand mental decline.
It really does.
Up until now, we've often thought of brain disease as simple physical damage, wear and tear or toxic buildup literally killing cells.
Just a mechanical problem.
Exactly. But this research suggests disease might actually be a failure of communication. The brain's waste isn't just trash, it's a daily intelligence briefing for the immune system.
I love that phrasing.
And when the plumbing breaks, the briefing stops and the immune system basically goes rogue. So let's recap this last bit. First, skull B cells sample brain waste to create immune tolerance. Second, inflammation causes leaky routing to the blood. And third, amyloid plaques cause internal clogs.
Spot on.
So to you listening, we often just think of brain disease as damage. But how does it change your perspective to know that disease might actually be a failure of communication between the brain's waste and its immune border guards?
It reframes neurodegeneration entirely. It is not just a failure of clearance, it is a fundamental failure of immune education.
So pulling this all together for everyone listening, right? Why should you care about the drainage of glowing green proteins in a mouse skull? Why does this matter when you're sitting at your desk rubbing your temples after a long, exhausting day?
It matters a lot.
Because this isn't just abstract biology. This is the fundamental hidden architecture of how your mind keeps itself intact.
Right.
Every day you engage in deep mental work, your neurons are firing, creating that mountain of metabolic trash, and your brain is carefully bagging it up, sorting it by specific local zip codes, and shipping it out to patches of your skull to gently train your immune system not to attack you.
It's an incredible balancing act.
And when that system is humming, you wake up the next day clear-headed. But when it falters, whether from a sudden inflammatory leak or a slow, chronic clog, the intelligence briefings stop. The fog sets in. And over decades, that loss of communication paves the way for diseases like Alzheimer's.
And if we look forward, the implications for medicine are staggering.
Oh, I bet.
I mean, we just established that the brain uses strict zip codes. The forebrain sends its waste strictly to the top of the skull. The deep brain structures send theirs to the base.
Or the nearest exit principle.
If that geographic mapping translates perfectly to humans, it opens up a revolutionary frontier. Imagine the future of neurology.
What could we do?
What if we could diagnose a highly localized brain disease, or even deliver targeted, region-specific drugs simply by accessing the exact corresponding square inch of the skull's bone marrow?
Oh, wow. You wouldn't even have to drill into the brain itself.
Exactly. We might be able to treat complex neurodegeneration by simply knocking on the right anatomical door from the outside, utilizing the local landfills to send signals back into the brain, completely bypassing the dangers of penetrating the blood-brain barrier.
That is an incredibly hopeful, provocative thought to leave off on. Treating the brain by simply managing the local immune training camps right outside its walls.
This is a whole new world of medicine.
So the next time your head feels physically heavy after a grueling day of thinking, try to give yourself some grace. Your brain isn't just tired, it's actively managing a wildly complex, zip-coded logistics network to keep the peace and keep you healthy for tomorrow.
Very true.
Thank you for joining us on this deep dive into the source material. Go let your brain clear its waste, and we'll catch you on the next one.